Hello, everyone. So my talk is going to be a little more focused on a single interesting enzyme, which is lysine deacetylase that regulates glucose metabolism. This is part of a serine hydrolase superfamily. Therefore, I would like to quickly run through the serine hydrolase superfamily. This is one of the largest enzyme families known in all three kingdoms of life, and it constitutes about 1% of the human proteome. It has over 240 enzymes, and as of now, 40% of the serine hydrolases are orphans, meaning that the biological substrate and the function of these enzymes are not known. Some of the famous examples of this superfamily are trypsin, chymotrypsin, fatty acid synthase, etc. And with over 240 enzymes, it participates virtually in all pathophysiological processes, including neurotransmission, metabolism, inflammation, cancer, and bacterial infection. And like I said, 40% of these enzymes are orphans. And one such enzyme is ABHD14B, which is an abbreviation for alpha beta hydrolase domain containing protein 14B. This is a human metabolic serine hydrolase enzyme. In 2004, it was reported to interact with the HAT domain of a transcription factor called TAF250, which is the largest subunit of the general transcription factor, TF2D. So this was very interesting. And back then, this enzyme was it was novel and nothing was known about this enzyme. And based on its amino acid sequence, it wasn't similar to any of the known eukaryotic transcription factors. So it got quite interesting. And the authors were a group of crystallographers. So they solved the structure of this enzyme, and from the structure, it was evident that this enzyme has a canonical alpha beta hydrolase fold and the conserved catalytic triad that a serine hydrolase has. So this triad is composed of a serine, histidine, and an aspartate. So these structural features suggested that this enzyme is a serine hydrolase enzyme. So to test if this has any hydrolysis activity, the authors have tested the hydrolysis activity of this enzyme on a generic hydrolysis substrate, which is a paranitrophenyl butyrate. And as you can see, CIB is another name for ABHD14B, was hydrolyzed by this enzyme successfully, proving that this enzyme is indeed a serine hydrolase enzyme, and it is hydrolytically active. And because of its interaction with the transcription factor, they also went ahead and investigated its role in transcriptional activation, for which they expressed a fusion of this enzyme with the DNA binding domain of Cal4. Expressed this protein in yeast cells that had a LAG-Z gene under the Gal4 element. Therefore, any transcriptional activation of this LAG-Z is an indication of transcriptional activation by this enzyme. And an expression of LAG-Z would result in blue colonies, and that's exactly what they saw when they expressed this fusion protein. This suggested that there could be a potential role for this enzyme in transcriptional activation. So this is the only report that existed about this enzyme when I started off with this project. Now, why are we interested in this enzyme? Like I just told, it interacts with the central component of the eukaryotic transcription machinery, suggesting that this enzyme could have a potential role to play in the transcriptional regulation. Apart from that, this enzyme has also been reported to be a prognostic marker of various cancers like gastroenteropancreatic neuroendocrine tumor, non-small cell lung cancer, and breast cancer, making it very important to study this enzyme. So I started off by asking the question of what are the biological substrates and what is the function of this enzyme. So to answer the first question, I performed the same paranitrophenyl substrate hydrolysis experiment, but just that I made a mutant of this enzyme to confirm that it is a serine hydrolase and not some other hydrolase like cysteine, aspartate, you have other hydrolases. So I mutated the predicted active site serine to alanine and tested for the hydrolysis activity using the PNP acetate as a substrate. And as you can clearly see, the wild type enzyme was able to hydrolyze the substrate, whereas the mutant was not able to do it, proving that this is a serine hydrolase, and the mutation at the active site serine renders the enzyme inactive. Following which, I performed a substrate profile where I used various PNP substrates with varying chain lengths of the carbon chain. So I used all the way from PNP acetate, which has a carbon chain length of 2, all the way up to C16, which is paranitrophenyl palmitate. And as you can clearly see, the enzyme was able to hydrolyze the shorter substrate the most, which is the PNP acetate. And with the increase in the chain length, the hydrolysis activity was dropping. This is also supported by the kinetic numbers for the 3 substrates that it was able to hydrolyze. And with the increase in the chain length, it is quite evident that the turnover rate, the affinity, and the catalytic efficiency was dropping, suggesting that this enzyme prefers smaller substrates. So what is the biological substrate of this enzyme? We know from the literature that this enzyme interacts with the histone acetyl transferase domain of TAF. Now, histone acetyl transferase domain, as we all know, uses acetyl-CoA as a substrate. And this enzyme forms a complex, and it has also been reported in the same paper that this enzyme forms a complex with acetyl-CoA. And from my own data, we know that this enzyme prefers smaller substrates and hydrolyzes PNP acetate the most. Therefore, our natural hypothesis was acetyl-CoA is the biological substrate of 14B. But through a series of experiments, which I'm not showing in the interest of time, we figured out that it is not acetyl-CoA, but CoA, which is the substrate. This is the same PNP acetate hydrolysis experiment, just that in the presence of CoA or acetyl-CoA, we are looking at the effects. So as you can clearly see, the hydrolysis rate or the hydrolysis activity is not affected in any way by the presence of acetyl-CoA. But in the presence of CoA, you see a significant increase in the hydrolysis activity. Therefore, we hypothesized again that following the hydrolysis of PNP acetate, the enzyme is probably transferring this acetyl group onto CoA to form acetyl-CoA as a second product of the reaction. That is exactly what we measured, and we found that the wild-type enzyme in the presence of PNP acetate and CoA was able to produce acetyl-CoA, which was not the case with the mutant enzyme or with no enzyme. Clearly proving that this enzyme is performing an acetyl transfer reaction and forming acetyl-CoA as a product. Now, PNP acetate is not a biological substrate, it's a surrogate substrate. So what is the physiological donor for this acetate group is the next question. So we again, through various experiments, figured out that it is the acetylated lysine residues of the protein that is the biological substrate. Here is a representational experiment showing you deacetylation of a diacetylated peptide by this enzyme. So instead of PNP acetate, I've used a diacetylated peptide, which is basically 20 amino acid long peptide that has 2 lysines that are acetylated. So we are basically looking at the deacetylation of this peptide and as a result, formation of acetyl-CoA. So it's very clear that all the diacetylated peptides are deacetylated to non-acetylated forms by the wild-type enzyme, whereas the mutant could not deacetylate the substrate. And as a result, the wild-type enzyme was able to form acetyl-CoA, which was not the case with mutants, which is something we already observed with PNP acetate as a substrate, suggesting that this enzyme uses acetylated lysine proteins as substrates biologically. Now, this is proved at in vitro conditions, but to validate it in vivo, I generated stable knockdowns of this enzyme in HK293 T cells. This is a Western blot showing you pan-acetyl lysine profiling of the 2 different knockdowns that I made for the enzyme and versus the control. And as you can clearly see, following the knockdown, there is a significant increase in the acetylation of proteins as compared to the control. And concomitantly, I also observed that there was a drop in the levels of acetyl-CoA in the knockdowns as compared to the control, suggesting that in vivo as well, this enzyme is deacetylating proteins and forming acetyl-CoA. Therefore, in the absence of this enzyme, you see an increase in the acetylated lysine levels and the drop in the acetyl-CoA levels. So therefore, the lysine, acetylated lysine containing proteins are the biological substrates of this enzyme. And this is quite interesting because the mechanism that is followed by the 2 well-known and well-studied classes of lysine deacetylases, which are HDACs and sirtuins are quite different from this mechanism that ABHD14B follows. HDACs hydrolytically cleaves the acetyl group from a lysine residue and releases it as free acetate, whereas sirtuins use NAD+ as a cofactor, and the hydrolytically cleaved acetyl group is transferred onto this NAD+ and released as acetyl ADP ribose. But in our case, ABHD14B is using CoA as a second substrate, and this hydrolytically cleaved acetyl group is transferred onto CoA to make acetyl-CoA. Now, acetyl-CoA, as we all know, is a central metabolite of glucose metabolism. Now that we know its biological substrate, what is its biological function? Before we dwell into that, I would like to just quickly run through the expression of this enzyme in different cells and tissues. So this is a confocal microscopy image of HK293T cells. And if you could focus on this green image, which is this cells, which is an image for the enzyme expressed in the HK cells. As you can clearly see, the enzyme is ubiquitously expressed throughout the cell, that is both in the nucleus and in the cytoplasm. This was true across different cell lines that I've tested for. And this is very interesting because the other two classes of deacetylases that we know, the HDACs and sirtuins, are known to regulate transcription, and they are present in the nucleus regulating transcription. So there is a possibility that this enzyme, which is present in the nucleus and known to interact with the transcription factor, could also be playing a role in transcription regulation. Sorry. I then looked at the expression of this enzyme in various tissues of mouse. And interestingly, this enzyme was highly expressed in liver and kidney. And we all know that these two are the metabolically active tissues in our body. And now we know that this enzyme is making the important metabolite, which is acetyl-CoA, and it is present in the metabolically active tissues. Therefore, this enzyme could also have a role to play in glucose metabolism or metabolism in general. So to find its function, the question that we asked is, could ABHD14B regulate gene transcription and therefore cellular metabolism? So to address this question, I first did a metabolic profiling of the knockdown cells and compared the levels of the metabolites with the wild type cells. For this, I performed LC-MS based targeted metabolomics of polar and non-polar metabolites. And this is a heat map showing you the levels of different metabolites that I've measured in the knockdown cells as compared to the control. And it's very evident from here that the majority of the changes are happening in the glycolysis and the TCA cycle. Now, to understand better of the most significant changes, this is a volcano plot showing you the significantly changing metabolites. Those are malate, pyruvate, succinyl-CoA, fumarate, all the intermediates of glycolysis and TCA cycle, which is something that I've represented in the pathway itself. So this is quite interesting, knowing that it is producing acetyl-CoA and knocking down the enzyme acetyl-CoA levels are going down. Now we know that it is also perturbing other metabolites in the glucose metabolism. Parallelly, I also performed transcriptomic studies to study the expression levels of genes in the knockdowns as compared to the wild type. And this is showing you the most significantly differentially expressed genes in red and blue. And when I performed a gene ontology analysis on these differentially expressed genes, it was interesting to see that the two major classes of genes that were differentially expressed were the transcription regulators and central metabolic enzymes. Now, this is quite interesting because this was the question to start with, whether the enzyme is able to regulate transcription and therefore metabolic enzymes or metabolism. So it was interesting to see this finding. And I did go through the central metabolic genes that were differentially expressed and figured out that there were quite a lot of genes that were part of glucose metabolism. To better understand these genes, I've put them onto their pathways that they participate in. This is a consolidation of both the metabolic and the transcriptomic studies. Here you have the different glucose metabolic pathways like pentose phosphate pathway, glycolysis, gluconeogenesis, TCA cycle and electron transport chain. Now, those in green are the upregulated genes. Those in red are the downregulated genes from the transcriptomic study. And the metabolites in the red boxes are the ones whose levels reduce significantly following the knockdown from the metabolomic study. Now, it is very nice to see that there is some correlation between the transcriptomic and the metabolomic study. That is, you see that the bisphosphoglycerate mutase is downregulated, and following that, you see a decrease in the levels of 3-phosphoglycerate. Same in the case of enolase 1, downregulation of which is probably causing the decrease in the levels of pyruvate and acetyl-CoA downstream. And even in the TCA cycle, you see that the isocitrate dehydrogenase 1 is significantly downregulated. And concomitantly, you see that there is a decrease in the levels of 2-ketoglutarate and succinyl-CoA. This correlation was very nice to see, but you need validation to bridge the gap. So all that we can say from these two experiments is that ABHD14B regulates glucose metabolism, which is possibly through transcription regulation. So you need additional studies to answer this. So what next? So ABHD14B, as we all know now, is a novel lysine deacetylase that is using CoA as a second substrate and forming acetyl-CoA as a result. Now, based on what this target protein is, the enzyme could have multiple functions. So the putative targets would be chromatin modifiers and transcription factors because it is present in the nucleus. It could be these two, and which is probably what is bringing about those transcriptomic changes that we see, which could then be resulting in the glucose metabolic changes and some other unknown functions. The putative target could also be metabolic enzymes, which could directly be bringing about the glucose metabolic changes that we see. And of course, you could have other proteins which could be doing unknown functions. So the third omic study would be a proteomic study, specifically acetylome investigation of these knockdowns to find what these target proteins are, which is going to probably bridge between the metabolomic and transcriptomic data and will help us validate the findings. So with that, I acknowledge all the people that helped me with the work and the funding agencies. Thank you. I'm open to taking questions.